those podocytes, those cells that make up the inner lining to Bowman's capsule, that have those little legs that overlap, create those gaps. Well, those gaps can change, right? You can stimulate those cells to squish up to make the gaps bigger, to stretch out and to make those gaps smaller. And that changes how easy it is for chemicals to filter across there. Okay. Everybody got this? So here's the diagram again showing, let's try to see this particular one though. The podocytes in purple, those purple cells that make up the lining of the Bowman's capsule wrapped around the glomerular capillaries. And so the gaps between these little branches on the cells can change. And depending on what the body needs, those gaps increase if you want to increase GFR, or the gaps can decrease if you want to decrease GFR. Changing that filtration coefficient. Questions about filtration? You guys got it? You have a question? Yeah.
I just think it looks insane.
Looks insane. Insane in a good way or a bad way?
Like in a really cool way, but like in
I see it as a really cool, complicated way that luckily we don't have to really think about in these words. Right?
I'm just amazed. That's bad.
It is pretty amazing.
Yeah.
Definitely. All right.
Yeah.
Let's move on to the next process then, reabsorption. What always blows me away when we think about all these complex processes that happen is it's amazing how often it actually works. Right? It's amazing we've made it as far as we have. Any one of these things can get screwed up. All right, reabsorption. So just to make sure everyone's got it in their head, reabsorption is going to happen with the rest of the nephron, between the peritubular capillaries and the rest of the tubule. And it's going to go in the direction of tubule lumen back into the body, so back into the blood vessels. Right? So holding on to chemicals. All right, reabsorption. Again, it's kind of like absorption that we saw in bulk flow, but there's a little bit more to it. And part of that little bit more is active transport. So absorption does not involve active transport, but reabsorption does. And that's why we don't call it just absorption. So there's active transport. We actively pump things back into our body, things we want to hold on to. And this is really primary active transport. And then there's also secondary active transport. Everybody remember the difference? Cool.
Oh, yeah.
Those are
Something
quick.
is being transferred by, so the first one, the molecule is being transferred, and the secondary active transfer is the molecule binds to something and that thing transfers the molecule.
Everybody agree with that?
Yes.
Kind of? So think about it more in terms of where the energy gets directly applied. So you've got this thing you want to move across a cell membrane, for instance. Right? If the energy from ATP gets directly applied to that thing that you're moving across the membrane and up its gradient, then that's primary active transport. If it's secondary active transport, your energy does not get applied directly to the thing that you're trying to move. Instead, you apply it to something else. Create a gradient, and now we can use that gradient to assist the thing we really want to move. Does that help? No?
Sorry, it creates a gradient, you said?
Yeah, so the example I think we used at the very beginning of the semester is, you can pump sodium across the cell membrane. Right, so you got a sodium pump, you use the energy from ATP, it moves sodium, you've got a sodium gradient. That's great. But if you want to move something else, let's say we want to move glucose. You could use a secondary active transport mechanism where you apply the energy to sodium to make a sodium gradient, and then you let sodium diffuse back across the cell membrane down its gradient that you just made. But when it comes back down, it grabs glucose with it. So the thing you're trying to move is glucose, but you didn't apply the energy from ATP to glucose, instead you applied it to sodium. So it's secondarily benefiting from that energy that's spent, the glucose is. Exactly what you said, right?
Yeah, so it's like grabbing a child and bringing him across the road, or you can go by yourself across.
carry the kid.
Yeah, like a newborn.
Well, sometimes you carry older kids. It's like, stop tying your shoes in the middle of the street, let's get going. You just pick them up and move them. Or something like that. Yeah, sure, just like the kid example. All right. Also with reabsorption, there is passive transport. Things can just take advantage of gradients that are there, and they can diffuse, for instance. And there's transcytosis. We talked about this a long time ago. Transcytosis, this would be endocytosis followed by exocytosis. Okay, so lots of different mechanisms involved in reabsorption. All right, looking at a little diagram here. So we're talking about the peritubular capillaries and the tubule. Reabsorption is going to go from the lumen of the tubule in yellow here, across the cells that make up the wall of that tubule, and then across the cells that make up the wall of the peritubular capillaries. It's going back into the body. Whether it's going passively or through primary, secondary active transport or transcytosis. All right, let's look at an example of reabsorption using active transport. So for instance, sodium ions. All right, so the sodium ions, if we're going to reabsorb sodium, we're going to try to go from the lumen of the tubule into the body. Well, we could go ahead and pump sodium from the cells that make up the lumen of the tubule into the extracellular fluid. And that would be an active process. So let's go ahead and draw some of this. Try to clear that up. So let's say these are cells that make up part of the tubule. So in here is the tubule lumen. On the outside here would be the extracellular fluid, but this is inside the body. So this is tubule in here. So we could go ahead and pump sodium right, from the cell into the extracellular fluid, right, towards the body. And then the next step is to go from the lumen of the tubule into the cell, and this would be a passive process. So once you pump that sodium out of the cell, you're decreasing the sodium concentration in that cell. You open up sodium channels on the lumen side, and now sodium can just diffuse right on in. So the active part is on the body side. Put the ATP in there to emphasize that it's active. So you pump it out on the body side, and then you allow it to diffuse in on the lumen side. But this whole process here is active, right? We're adding energy to it to move sodium from the lumen of the tubule into the body. That's reabsorption.
So, sorry, I'm thinking of my question. So for filtration, it goes through also the cells, or does it go through the gas in the...
Depends on the chemical, so it can do both. If it's a gas like oxygen molecule, it could go through the cells itself. But most of it's going to go through all those gaps. It's really leaky.
Yeah.
Right? But again, that filtration, that's going the other direction.
Right, right. So then with absorption, or adsorption, reabsorption, it's the same thing. It just depends, or it
It's
always
not
goes
going through the gaps because we don't have those big gaps in the rest of the tubule.
Yeah.
There's more control over what goes in and out.
Right.
Remember with filtration, it's kind of blind, like just let everything go,
Right.
except for cells and proteins. But now we're going to be real selective in terms of what we reabsorb and what we secrete.
I see. So that's why the place where reabsorption occurs is very specific.
Yes, and so yeah, reabsorption along the entire length of the nephron is not the same.
Right.
It's going to vary as you go along.
Got it.
Okay, anything else for now? All right. Here's a figure that shows the same thing. So we've got on the left is the tubule lumen, on the right is the body. So we're going to pump sodium from these cells that make up the wall of the tubule, yes, into the body. In this case, we're using the sodium-potassium pump. And then we just open up sodium channels on the lumen side. Sodium diffuses down its gradient. Right, how did we get the low sodium concentration in the cell? Well, we pumped it out. Right, we just have to put the pumps on the body side, so we get net absorption. Sorry, net reabsorption. Okay. All right, let's see an example of secondary active transport. Guess I could make this more complete. Let's turn this into a sodium-potassium pump. All right, for secondary active transport, we can use that same sodium pump. So we can pump sodium from the cell towards the body, the extracellular fluid. That's an active process. And then on the lumen side, we can have some facilitated diffusion. But in this case, it lets sodium go through, but it also requires glucose to be present. So this protein here is not going to let sodium go through unless glucose accompanies it, goes with it. So what are we moving by secondary active transport here? We're moving glucose. Where do we apply the energy? To something else, in this case, to sodium. We apply the energy to make a sodium gradient, then we use that sodium gradient to pull glucose into the cell. And then we can let glucose just pass from the cell to the body side, and it's going to go down its gradient there. All right, so you keep pulling glucose in with secondary active transport, that raises the glucose concentration in the cell. Now glucose can just diffuse down its gradient towards the body. Why doesn't glucose build up in concentration out here? Because you've got a blood vessel here, right, where fluid's flowing through and it keeps picking up that glucose into the blood and gets carried away. So that blood maintains a relatively low glucose concentration on this side. Questions about that? Yeah.
How does the glucose go from the cell to the fluid after?
Number 3 there?
Yeah.
So that's going through by, there's a carrier protein in the cell membrane that allows it just to go down its gradient there. So, are you getting tripped up by the secondary active transport? So really, what is secondary active transport? It's this whole thing together. It's glucose's journey from one side of the cell to the other. Right? And so the goal is, let's get it from the lumen to the extracellular fluid. How do we get it there? We use the sodium gradient. And it was 2 steps. We had to go through cell membrane both sides. So in a way, that's sort of misleading to suggest that this whole thing, just step 3 is secondary active transport. It's really 2 and 3 together. Right? We're not applying energy directly to this step. We're not applying it to this step either. The energy got applied at step 1.
Makes sense.
Okay? Anything else for now? All right, here's a figure that summarizes this. So what do we have? On the left side, we have the lumen of the tubule. On the right side, we've got our peritubular capillary. The blue in the middle here represents the extracellular fluid. We're going to go ahead and, I should point that out. Where you see red arrows, that's an active process. Green arrows represents a passive process. So this little circle here is a sodium-potassium pump. It actively pumps sodium out of the cell, and it actively pumps potassium into the cell. By doing this, removing the sodium from the cell, we decrease the sodium concentration in the cell. And now sodium can passively just diffuse into the cell from the tubule lumen, going down its concentration gradient. But this particular protein will only let sodium go through if glucose goes with it. So here, glucose's arrow is colored red because this is active. But it's not primary active transport. We're not using ATP right here. It's using the energy that we invested in sodium to pull it along into the cell. And once it gets into the cell, it raises the glucose concentration in the cell. That glucose can then diffuse down its concentration gradient into the extracellular fluid and then into the blood vessel. So glucose traveled by secondary active transport here. Sodium was pumped by primary active transport. So really we have both examples in this picture. How do you guys feel about that? Just as excited as you've been all class? Oh my gosh. Okay.
I have a question.
Yeah.
Is the lighter purple? Is it purple? I don't know. Does that represent?
Right here?
No, sorry. For the glucose that is pumping out or diffusing
Oh,
out?
here versus here?
Yeah.
So why is that colored differently?
Is it just a different protein or is it like a channel?
They ran out of some of that colored ink.
Oh, right. Yeah.
I don't know. Why would that be? Maybe they're suggesting that everything about this is passive, where there's some active components to these. I don't know.
Okay, I just wasn't sure if it was like.
That part's not on the test.
Okay.
Anything else? All right. What about passive reabsorption? So we talked about active, primary active transport with reabsorption, secondary active transport with reabsorption. Now let's look at passive reabsorption. So if it's passive, we're not going to add any energy to it. We're just going to rely on gradients that are already there. So a chemical that does get reabsorbed passively is urea. It'll diffuse from the lumen of the tubule back into the extracellular fluid. And at that point, it can get taken up into the peritubular capillaries. So that urea gets concentrated in the tubule lumen. As that fluid passes through the tubule, we reabsorb most all of that fluid, leaving behind the urea. That urea gets more and more concentrated, and then by the end, when you get to the collecting duct, the urea in the tubule lumen is fairly concentrated, and that can get reabsorbed passively. Which sounds a little weird. I'm getting some strange looks from people right now. Because urea is a metabolic waste product. If it's in the tubule lumen, it's where you want it to be. Leave it there. Let it go out with the urine. But we'll see later on that there's an instance where we do reabsorb a little bit of that urea, and there's a benefit to it. And we'll talk about that when we get to it next week. Okay. All right. So what do we have here? So here's our simplified nephron. We've got all this filtration. Lots of reabsorption occurs down the length of the tubule. And as we reabsorb that water, urea gets more and more concentrated. So when we get down to the collecting duct, urea is at a fairly high concentration relative to the body and what it started at. So now we have a high urea concentration in the tubule lumen, relatively low urea concentration in the body. Urea can just passively diffuse down its concentration gradient back into the body. So that's an example of passive reabsorption. Okay, questions about that one? All right, and then our last example, transcytosis with reabsorption. So I mentioned in the beginning that with filtration, you've got plasma, except for proteins, it gets filtered. Some proteins are actually filtered, some sneak through. So you can reabsorb those with transcytosis. And remember, this involves vesicles. You've got endocytosis on one side, exocytosis on the other, and that involves the making of, the using of vesicles. Okay. All right. Let's go ahead now and talk about transport saturation. All right, so we've been transporting all these chemicals with reabsorption. Can you transport at an infinite rate? No, things can get saturated. You can only work so much. So let's see how this would work. So the rate that you can transport chemicals, that's going to depend in part on the concentration of the substrate being transported, whatever that chemical is. It's also based on the number of transports. So today you're trying to reabsorb some chemical. Maybe you're trying to reabsorb glucose. Well, the more glucose available, the higher the transport rate. If there's very little there, you're not going to transport it at a very high rate. Right? So the concentration of the substrate is important. Also the number of transporters. Let's say you only had one transporter for glucose. Well, you're pretty limited. You're not going to be able to move a whole lot of glucose at one time. If you had bajillion of these transporters for glucose, you could transport at a much higher rate. So the more transporters available, the higher the rate of transport you can get. Let's look at a graph here to get an idea of some of this might work. So we're trying to transport some substrate. And if we look at the concentration of that substrate in your blood plasma, use that as the horizontal axis, and then the rate that that substrate gets transported, let's say reabsorption, we can go ahead and graph that out. So the more substrate you've got in the blood, as that increases, the rate of transport, reabsorption, also increases. There's more available, you're going to reabsorb more of it. Right? So you've got a higher concentration, let's say, of the substrate in your blood, you filter more of it, there's more in the tubule lumen, you can reabsorb more of it. And that's what we see with this relationship. Okay? Plasma substrate concentration goes up, the rate of transport goes up. To a point. And at this point, where you no longer increase the rate of transport, you've reached saturation. So all the transporters are working as hard as they can, and even if you add more substrate, you can't move it at a higher rate. Everything's working as fast as it can. Right? So let's say we had lots of doors at this end of the classroom that led out into the hall. And we wanted to empty students out of the classroom. Right? The doors are the transporters. Right? If there are only 2 students in here, okay, well, you could get those 2 students out and it would go at some rate. Right? But it's not a real high flow rate of students. Right? Let's say you've got 60-something students in here. Right? Now you've got a higher concentration of substrate. You guys are all substrates now. Right? And so we're going to have a higher flow rate out into the hall. But if we only have 2 doors, that's going to restrict the rate that everybody leaves the classroom. If there were 20 doors up here, then we could leave at a much higher rate. It's the same idea. Right? All right, let's go ahead and apply this in a slightly different way, and we can see how this influences some of the other processes involved. So the substrate here we're going to be looking at is glucose. Right? So we could track plasma glucose concentration on the horizontal axis and a number of aspects of glucose on the vertical axis. The blue line represents the rate that glucose gets reabsorbed, taken from the tubule lumen back into the body. The orange line, which actually goes under the blue line here, that's the rate that glucose gets filtered from the glomerulus into Bowman's capsule. The green line represents the rate that glucose gets excreted, that it leaves the tubule into towards the urinary bladder. This black dotted line represents the maximum amount that you can actually transport. All right, so let's follow the orange line. The more glucose that's in your blood, the more you filter. Right? You've got the same concentration of glucose in your blood plasma that you have in that filtrate in Bowman's capsule. Right? And that's why we get this nice linear relationship all the way up. And at least in the beginning, the more glucose you have in your blood, the more you filter, the more you reabsorb. In fact, for this first portion here, all of the glucose that you filter, you reabsorb. That's valuable energy. You don't want to just pee out sugar. Right? You want to hold on to that. And under normal situations, you do. You reabsorb all of that glucose. To a point, though. If the amount of glucose in your blood is really, really high, then your nephron is not going to be able to keep up with all that extra glucose. So you're going to reabsorb as much as you can, but the extra glucose that got filtered actually now is not getting reabsorbed. Instead, it starts to get excreted. So once you've saturated all your transporters, Now some of that glucose passes through the tubular lumen, not getting reabsorbed, and it gets excreted, which means it goes out with the urine. You see how that works? But in most situations, your blood glucose levels do not go above 200 milligrams per 100 milliliters. Right? So keep it down in here. But you drink, 4 Jolt Colas and 7 Twinkies, then it sends your glucose levels up a lot higher when you start peeing out sugar. Questions about any of that? All right. Let's look at some of the pressures involved. So part of reabsorption is absorption. All right, remember, we didn't get net absorption at the glomerulus, but we do get that at the peritubular capillaries. So what are these pressures involved here? To figure out what's driving absorption here, we need to look at the pressures. So the hydrostatic pressure of the capillaries, the blood pressure in those peritubular capillaries, is pretty low at this point. It's down to around 10 millimeters of mercury. Remember, it was around 60 millimeters of mercury in the glomerulus. Once you go through that glomerulus, though, you remove 20% of that volume, that pressure is going to drop a lot. So it's down to around 10 millimeters of mercury in the peritubular capillaries. That osmotic pressure in the capillaries is still at around minus 29 millimeters of mercury, so it stays high. So these are the main pressures involved. What's the net pressure? Minus 19 millimeters of mercury. So that minus tells you that that's an absorptive pressure. And this explains why you do not get filtration at the peritubular capillaries. The blood pressure is too low compared to that osmotic pressure. Okay, questions about that? All right, next process. Secretion. This one's pretty easy. So remember, secretion. This is our second opportunity to get rid of chemicals. It's going from the body into the tubule lumen. Most secretion is through secondary active transport. Phew, that was tough. All right, next process, excretion. Excretion is pretty simple also. So remember, all this fluid going through the tubule is considered filtrate. And you're modifying its contents as it goes through, all through the nephron. But once this filtrate leaves the collecting duct, it's done being in the nephron, it enters into the renal pelvis, down the ureter to the urinary bladder. It's no longer filtrate, and at that point it's considered urine. So urine leaving the collecting duct is the process of excretion. Okay. All right, so that filtrate went through the nephrons and the kidneys. When that filtrate leaves the kidneys, it goes through the renal pelvis, that's excretion. And now it's urine. And it's just going to trickle down the ureters into the urinary bladder where it's stored until you're ready to get rid of it. And keep in mind that as that filtrate goes through the renal pelvis, the ureter, the urinary bladder, and urethra, all of those parts, you do not modify its contents. It's as good as gone. You still have to carry it around with you. Okay? That's not true for all vertebrates, though. There are some, like for instance frogs, that can do some reabsorption in the urinary bladder. They can, if they need some of that water that's in there, they can reabsorb some of the water through the wall of their urinary bladder, which is pretty handy, right? Especially if you got to maintain wet skin, because you're losing water all the time. But we're not frogs, so we don't get to do that. What's that? Womp womp. Yeah. All right. So now we've got urine in the urinary bladder, and it's being stored in there. The bladder's filling up. As I talk more and more about this, more and more people have to go to the bathroom, right? So now we're ready for our last process, micturition. Who actually was able to use this word in the conversation? No one tried, did you? You should try this. All right, so what is micturition? This is voiding urine from the bladder. Emptying the bladder. So why doesn't urine just pour right on through the bladder on its own? Because it doesn't. That's because there are valves that control urine flow out of the bladder and through the urethra. And there are two valves involved. The first one is the internal urethral sphincter. So what's a sphincter? The name we all laughed at when we first heard it in grammar school and middle school. Why is the word sphincter funny? You guys admit it's a funny word, right? So, the sphincter, it's a circular muscle around a tube. When it contracts, it squashes that tube and closes it off, acting as a valve. So the internal urethral sphincter, this is a ring of muscle right at the very beginning of the urethra, at the base of the urinary bladder. And this sphincter is composed of smooth muscle. Smooth muscle. So you do not have conscious control over the contraction or relaxation of this valve. It's out of your control. Okay. The second valve, probably guess what it's called. The external urethral sphincter. And lucky for us, this one is composed of skeletal muscle. Right, so it's further down in the urethra. And what's the benefit of it being made of skeletal muscle?
We have control of it.
We potentially have some influence over when it contracts and relaxes. Right? A learned skill. All right, so skeletal, this one's made of skeletal muscle. We can control when it contracts and relaxes. All right, let's go ahead and look at the setup here. So on the left, we have the male system. On the right is the female system. In terms of controlling micturition, all the same parts. So we've got the urinary bladder up top, and these two little horns that stick out are actually the ureters. So those are coming in from behind, come around underneath the urinary bladder, and these little dots here represent where those ureters drain into the urinary bladder. And your kidneys, remember, when everything's working correctly, are constantly filtering blood, constantly making urine, it's constantly trickling in to the urinary bladder. And then out of the bottom of the urinary bladder, we've got the urethra that runs right through the floor of the pelvic cavity. At the very base of the urinary bladder, at the beginning of the urethra, that's where that internal urethral sphincter made of smooth muscle is. The external urethral sphincter is made